Start. Okay, it's 7:12 now. All right, so let's begin. Right, so I'm doing depression and anxiety. There's not much to know for anxiety this year because she took out most of the content. So we'll just run through it briefly at the end. Okay. Right, so we'll start with depression. These are your learning outcomes from Dawn. So we'll begin with the first one. So etiology of depression. As you guys probably already know, depression is a multifactorial disease, and it comes as a result of an interaction between the individual and their environment. So I've divided it into external factors that can lead to depression and internal factors. So in terms of external factors, it's pretty obvious. It's life circumstances like being lonely, bullying, and is there any way I can get rid of that thing? No, it's fine. Okay, yes, and bereavement in the family. In terms of internal factors, I don't know if you guys have heard about the cognitive behavioral model, but essentially it states that cognitive, behavioral, emotional, and physiological states within a person can lead to depression. So in terms of cognition, having feeling of a lack of control over your life can lead to that. Behavioral, staying in bed, missing deadlines and appointments can contribute to that. Physiological changes are more the neurological basis of depression, as well as like long-term illnesses and things like that. And emotional states, essentially, I guess that's dwelling on feeling sad and things like that. Okay. And so now we'll move on to the neurological basis. Part of the internal environment that leads to depression. So I've separated neurological basis into structural changes and physiological changes, but obviously these distinctions don't actually exist. It's just to simplify your understanding of it. Okay. And what I mean by that, sorry, is that physiological changes normally lead to structural changes. Okay, so in terms of structural changes, we tend to think in terms of depression that there's a reduction in brain mass in areas like the prefrontal cortex and the hippocampus, and that leads to a resultant decrease in neuronal activity. And this is just an example of that, where you can see a bit of atrophy in the frontal lobe and near the temporal region where the hippocampus is supposed to be more inside. And there's also an increase in brain mass and activity in places like the amygdala and hypothalamus. So as you probably know from your anxiety lecture, the amygdala is more involved in fear responses, amongst other things. And the hypothalamus hosts the pituitary gland, which releases hormones that result in an increase in cortisol. Okay, now focusing more on the physiological changes. Okay, so the main one is the neurotransmitter / monoamine theory of depression, which essentially states that reductions in serotonin, noradrenaline, and dopamine levels can lead to depression. Okay, so this is just what Dawn put in her lecture, so I'm just going to briefly summarize it. So a big serotonin hub in the brain is the raphe nucleus, and it projects into the cortex, down into the periphery, and within the brainstem. But really, you get serotonin projections in quite a lot of places within the brain. And so those projections project to areas involved in mood, nausea, vomiting, sleep, and cognition. So as you can imagine, alterations in those projections / how much neurotransmitter they're getting can lead to symptoms of depression. And peripherally, serotonin affects GI motility and hunger, obviously. Okay, in terms of noradrenaline, in terms of noradrenaline, one of the big hubs for noradrenaline is the locus coeruleus, and that also projects centrally into the cortex and peripherally, as well as the cerebellum. And as you can imagine, alterations in neurotransmitter levels to these areas can also lead to the symptoms of depression. And this is just to summarize that. Okay, so next is the neurohormonal theory of depression. Essentially, the take-home message from this is that prolonged increased levels of cortisol within the body can potentially lead to neuronal damage and brain atrophy. And those areas are notably the temporal lobe, which hosts the hippocampus, and the prefrontal cortex. So increases in cortisol can come as a result from like prolonged steroid use, like in Cushing's, and chronic stress, which Ben talked about in his neuroendocrine lecture, the HPA axis, essentially. So those can lead to those brain changes. Okay, immune theory, quite briefly. One is brain inflammation. So it's thought that either the depression can lead to neuronal changes that cause inflammation, which make things worse, or there's existent neuroinflammation that can lead to neuronal cell death. And the gut-brain axis, which has become popular in the last few years, it's essentially stating that the flora in your gut can lead to feedback to the brain, which can potentially cause symptoms of depression. And interestingly, in animal studies, they've done fecal transplant in animals that have the depressed phenotype, and they've replaced it with rats' feces of a normal phenotype, and they've saw behavioral changes. So that's super interesting. Okay. Right, neuroplastic changes of in depression. Essentially, this just states that generally in depression, you see neuronal changes, so changes in the dendrites of the neuron where there's a reduction in arborization, and there's a reduction in synapses and spines on the actual dendrites. Not really sure whether that comes as a result of depression or whether that leads to the etiology of depression, but it's an observation that's generally noted. Okay, so now we're going to move on to the symptoms of depression, and the symptoms will depend on what type of depression you have. And the two main ones that Don focuses on is unipolar depression, which is persistent and chronic low mood. And, sorry, I just wanted to make a note here to say that where we diagnose depression is slightly arbitrary, which only means that a group of people who made the DSM 5 and the ICD-11 have decided that there's a threshold after which you diagnose depression in terms of how many symptoms and the severity of the symptoms, and below that threshold where you won't diagnose. But if someone is constantly below that threshold of a depression diagnosis, that's called dysthymia, and it can be what's also known as chronic mild / subwhole depression. But the diagnostic, a diagnosis of depression can be made when the symptoms cross that threshold set out by the ICD and DSM, which we will go into a bit more detail on in the next slide. In terms of bipolar depression, that essentially just comes about when a person has major depression, but they also have periods of mania, which is essentially a sense of euphoria. They're very active, they're impulsive, they tend to do things they wouldn't normally do, essentially. And it's the same idea in terms of the threshold. So over a certain threshold, they're considered manic and depressed, and under that threshold, they're considered cyclothymic if they're oscillating between periods of low mood and euphoria. And a diagnosis of bipolar disorder is made when their symptoms cross those thresholds. Okay, so in terms of symptoms for major depression, the key features is that the person has to have these symptoms for more than 2 weeks, and they have to have persistent low mood and anhedonia, as well as other symptoms of depression, which can be cognitive, behavioral, and physiological. I like to use the mnemonic Leeswig. So low mood, changes in eating, anhedonia, sleeping, energy, worthlessness, inability to concentrate, and guilt. So all of these include the DSM criteria for depression, and it's just an easy way to remember it. Okay, And for the DSM, you have to have 5 of these symptoms for more than 2 weeks to get a diagnosis of depression. And for the ICD, it's 4 symptoms for more than 2 weeks. Okay. Bipolar disorder, the DSM states you can diagnose bipolar disorder when they have major depression and when they have had one manic episode. The ICD states that you need recurrent episodes of mania and depression, and you can treat it with lithium. Okay, so in terms of depression management, NICE sets out a stepped care approach to treat depression, moving from more mild cases to more severe cases that are treatment resistant. So in the beginning, when someone comes to the GP and you're not sure whether they're depressed, you need to see whether their symptoms last for more than 2 weeks. And then after, you can consider a diagnosis of depression and whether you want to treat them. So given that they have been depressed for 2 weeks or more, you can make a referral to the IAPT service where they will offer low intensity CBT. So meaning cognitive behavioral therapy. So if that doesn't work, you step them up to high intensity cognitive behavioral therapy, and you can add pharmacology if you feel it's appropriate. If those both don't work, you need a specialist referral, and they will offer more advanced interventions like transcranial magnetic stimulation and transcranial direct current stimulation, which we'll go through in a minute. And last but not least, your last line of defense against depression is electroconvulsive therapy. And this is the gold standard of treating depression because of its outcomes, but it's also the last line because it's the most invasive. Okie dokes. Right, so I'm just going to explain the CBT model real quick because you guys have had a lecture on it as well. So it's essentially, like we were saying in the beginning, a relation between your thoughts, behaviors, bodily sensations, and emotions. So I'll give an example of that. So let's say you're on your way to developing depression and you start waking up in the morning and staying in bed instead of engaging in your normal day-to-day activities. You might start to feel low in mood. You might start to have thoughts of being inadequate, being worthless. You might start to think, oh, why can't I do anything right? And then that might lead later to feelings of fatigue, tiredness, lack of motivation. And you can see how this cycle can just go round and round and round, essentially. What cognitive behavioral therapy tries to do is to break this cycle in one of these domains. So for example, in therapy, they try to do things like thought challenging to show you where your thinking is faulty, and as a result, how you can create more adaptive thoughts in placement of those maladaptive thoughts. In terms of behavior, they use something called behavioral activation, where they essentially make you create a schedule and they ask you to follow the schedule instead of your mood. So that pulls you out of those maladaptive behaviors that lead to this vicious cycle. In terms of bodily sensations, this is interesting because sometimes you can interpret things as fatigue or high heart rate to mean that you're depressed or that you're really anxious, when really that's just a physiological response that's going on and it's a misinterpretation. So for example, if you give propranolol, which is a beta blocker, you can intervene at this level, which can break that vicious cycle as well. Okay. So mild cognitive behavioral therapy, so low intensity CBT, is essentially for mild to moderate depression and anxiety. And it's where they give you a guided self-help book or group therapy so that you can work through breaking the cycle on your own with a catch-up session with one of the therapists once a week. So if that doesn't work, we need to move to high-intensity CBT and pharmacology. So this is where you sit down with a registered cognitive behavioral therapist for 1 hour a week, and you really go through the things that are keeping you in the vicious cycle, and you try to make concrete plans to change those behaviors. So this is for moderate to severe depression or anxiety, or it's when the previous step has failed, like we said. Yep, it's an hour a week. And you can add pharmacology at the depression of the psychiatrist, or sorry, at the discretion of the psychiatrist at the service. Okay, so I'm going to do a brief segue into pharmacology because it's on your drugs list. Okay, so first line treatment for depression is selective serotonin reuptake inhibitors, and this is how they work. So serotonin is released into the synaptic cleft, causing depolarization of the postsynaptic neuron. So SSRIs block the reuptake of serotonin into the presynaptic terminal, leaving it to have its or a greater impact on the postsynaptic neuron. An example of these on your drugs list is sertraline, fluoxetine, which is Prozac, which was in the picture, and citalopram. Indications obviously are depression and anxiety, and because Dawn put this as a learning outcome, some of the side effects are insomnia and nausea. Okay, so monoamine oxidase inhibitors. So these inhibit the breakdown of the monoamines. Remember the monoamine theory of depression that we were talking about. So mainly serotonin, dopamine, and noradrenaline. So as you can see here, monoamine oxidase is part of the breakdown of dopamine in this pathway. And if you block monoamine oxidase, you get an increase in dopamine and noradrenaline. And an example of this is meclobamide, and causes the cheese reaction. Sorry, I know that cheese is spelled wrong. Sorry about that. Okay. Tricyclic antidepressants. So one way to remember the mechanism of action of TCAs is ham, no shit. So ham will be H1 antagonism, adrenergic antagonist, and muscarinic, so cholinergic antagonist, noradrenaline reuptake inhibitor, and serotonin reuptake inhibition. Example of these are clomipramine, amitriptyline, and nortriptyline. And one of the big side effects that you need to remember for anticholinergics is anticholinergic syndrome. So it's essentially antagonism of the parasympathetic nervous system. And as you can imagine from first principles, this might look something like mad as a hatter, red as a beet, blind as a bat, because remember, the parasympathetic nervous system causes vasoconstriction in the eyes. So if you have unopposed dilation of the pupils, you won't see as well. And dry as a bone would be anhidrosis, and hot as a hair would be an increase in temperature. Okay. So other antidepressants, they work similar to the SSRIs. They're just different monoamines. So noradrenaline reuptake inhibitors are one class. So that's reboxetine. Serotonin and noradrenaline reuptake inhibitors, that's venlafaxine. Dopamine and noradrenaline is bupropion. And you also have alpha-2 adrenergic antagonists, which is mirtazapine. Okay. Okay, so we've been through the first 3 stages. Now we need to make a specialist referral. So I'm going to quickly touch on TMS and TDCS and ACT, just because she talked about it a little bit. I'm not sure if she explained it much in her lecture. Okay, so to explain TMS and TDCS, I need to briefly explain electroencephalography, so EEG. Okay, so essentially, this is when you put a very attractive cap of electrodes onto a person's head, and you can create conductance through the skull by adding a layer of saline gel into these little pots. And then that can create a current, which essentially picks up activity from the skull, essentially. And each of these electrodes will pick up a population of neurons that fire. So this is a better way to visualize it. So let's say these electrodes are picking up activity from this part of the skull, and then those ones are picking up activity slightly to the right. and so forth and so forth. Okay, so what transcranial magnetic stimulation is supposed to do, it's supposed to activate a certain population of neurons in a certain region of the brain. And it's thought that activating certain populations in certain regions can lead to alterations in brain signaling, which can, as a result, cause changes to the symptoms of depression. Okay, and transcranial direct current stimulation is just a slight variant of TMS in that in TMS, you tend to induce activation of a certain population of neurons, where in TDCS, you just tend to modulate neuronal activity rather than creating activation per se. So it's the only difference is that transcranial direct current causes lower level stimulation than TMS, basically. Okay, and the gold standard, ECT. So essentially what you're doing here is you're trying to induce seizure-like activity in certain areas of the brain so that it can change the chemical environment within that brain region. And that's supposed to, and that change in physiology and environment in that brain region is supposed to work kind of like a reset button. So if a person's neuronal circuits are firing in a certain way and they're constantly ruminating about certain things, if you change the environment there, that's supposed to change the activation of those neurons and pull them out of those states that are keeping them depressed, basically. Right, anxiety. So I'll briefly go through the neural circuits related to anxiety, like Dawn wanted. Okay. So the take-home message from what Dawn is saying, because I know it can get quite convoluted sometimes, is that in anxiety disorders, input and output to and from the amygdala are strengthened. So this means that, okay, so if you have an increased strength of input into the amygdala, this means that you are more able to detect like low-level threats in the environment, since the amygdala is involved in fear responses, right? So if you can imagine, if you have a strengthened input, you're going to be more vigilant about things that cause fear responses. And as a result, you'll be more easily scared, essentially. And in terms of output, if that's strengthened, then since the amygdala projects to so many regions, including the sympathetic nervous system, you will have a physiological fear response more easily as well. Okay, so very briefly, the neuronal structures involved in anxiety. Obviously, you have the amygdala, which is involved in fear, social recognition, and olfaction. Then right beside it, the hippocampus, which is part of the limbic system, prefrontal cortex for higher order functioning, the insula, which can be seen if you separate the temporal lobe like this, and the association area, so parietal and frontal. And then obviously your sensory motor and autonomic regions like we just discussed. Okay, neuronal mechanisms involved in anxiety. So like I was saying before, because you have an increased strength of input and output to and from the amygdala, you have an increase in CNS activity, increased neuronal synchrony, especially within the amygdala, which means a lot of neurons are firing together more readily. Firing frequency changes, which means these neurons are firing more frequently, which means there's a strengthened output, and morphological and genetic changes. So obviously, if the amygdala is more active, there's going to be morphological changes within the amygdala. And one nice way that Dawn put it here is that, so here you're looking at differences in an increase in metabolism within the amygdala. So this is just a comparison of a control to a person with PTSD. And you can see here that there's increased activity in the amygdala versus a normal person in PTSD, social anxiety, specific phobia, and learned fear, basically. And last but not least, brain output changes. So the amygdala projects to all of these different areas, And since the output activity is increased, you can imagine that these target areas will be more readily activated. And as you can see, a lot of the time it's involved in sympathetic activation. Okay, well, that's it. Thank you very much. Please scan our QR code to evaluate how this morning has gone for you. It helps us with our portfolios. And yeah, let me know if you have any questions. I just added some extra notes on CBT in case you guys wanted it as well. Thank you.